Hello, thank you for inviting me to participate in this workshop. I'm excited to be here and share my personal opinions and research regarding the building of a taxonomy for consistent determination of design differences in combination products. Before I begin, just a little background about who I am and where I work. I'm Mary Beth Privatera, principal of HS Design, which is a StereoPack company that focuses on user-centered design. We work across the practice of medicine with over 40 years of experience in the field. My background is in design. I'm formally trained in industrial design with a PhD in design and have always been involved in the application of human factors in design. I'm currently the co-chair of AMY's Human Engineering Committee, and I firmly believe that usability happens or doesn't happen as a result of design. The research I'm presenting today is a result of FDA-funded efforts with the excellent research team, including Dr. Megan Conrad, Melissa Lemke, and Dr. Molly Story. Each of us bring unique strengths to our research program and have a successful history of collaborating on many different efforts throughout my history and of involvement with AMY. Please note the research I'll be presenting today does correlate and build upon the previous presentation by Melissa Lemke. She covers AIMS 1 and 3, and I'll be covering AIM 2. To reiterate from Melissa, our FDA-funded research is to identify and analyze user interface design differences that may impact substitutability of an RLD and proposed generic drug-device combination product for clearance through the FDA and a pathway, in consideration of the needs of key stakeholders, the end users, FDA reviewers, industry, and academic researchers. As Melissa covered AIMS 1 and 3, my goal is to dive deeper into AIM 2. AIM 2 is the development of a visual taxonomy to systematically analyze user interface design attributes and identify minor and other design differences. They say that a picture is worth 1,000 words, and we're going to find out if that has application for comparative use human factors. Let's start with the guidance. The guidance requests a completion of a threshold analysis and, as a result, a determination of design differences that are possible. They list a few options in describing the design differences. 1. No design differences, when no differences are identified between the reference and the generic. Differences in design, if differences are identified between the design of the user interface of the proposed generic combination product and that of the RLD. If present, these can either be minor or other. Minor design differences mean that the design difference does not affect any external critical design attribute. Other is when the differences in the design of the user interface may impact external critical design attribute that involves the administration of the product. The impact of this determination is that FDA may request applicants provide additional information or data, such as data from a comparative use human factors study, and to address the differences identified in the user interfaces and that may introduce a risk of impact to the clinical effect or safety profile of the generic product. Further, the guidance recommends that design revisions that minimize differences from the RLD. This means that if you've made improvements, then you may want to go back and rethink those improvements because now you've got a little bit more information to provide. Sorry about that. The goal in building a taxonomy of design is to really be able to more consistently identify design differences and to just help clean up that world between what is a design difference, does it matter, is it minor, is it other? Is there a design difference? And thinking about design differences, let's take a simple example shown here. This is an example of a side-by-side comparison. It's only the first part in determining design differences, so let's take a stab at it. To the untrained eye, these could look exactly the same. They're all gradations along the same rectangle. However, to the trained eye, these are all three different. They vary in size, color, and shadow. In assessing the design differences between a proposed generic and an RLD, it's important to determine not only what design differences exist, but also whether or not they matter. This should rely on the use-related risk analysis and are recognizable by the user group. And lastly, that they do not impact safe and effective use. The goal of any design related to a combination product is to enable accurate dosing in a safe, effective manner. This often requires a reduction in complexity. So design matters. These facts are driving the need for AIM 2 of our research. The fact remains that the distinguishing between designs depends on the context and the person. Context matters because it could be variable. It could impact user expectations dependent upon their environment. It could also impact other attributes such as access to care. Furthermore, the user group characteristics matter because this directly impacts their abilities to interpret design. Ultimately, this may influence device use. Determining design differences of the physical design can be evident, however, it is the interpretation of the user interface, including labeling and training, that matters. In the case of a proposed generic versus RLD, design interpretation will largely be driven from the previous use of the RLD. However, naive users may interpret the design through exploring the product and its features with reliance on their personal expectations of functionality, previous experience with like items found in the consumer world, therefore building mental models of how a device is intended to be used and have further expectations when that context changes. This speaks to the importance of robust and detailed task analysis. So specifically talking about AIM2, our goal is to build a taxonomy using RLDs and use-related risks as a backbone for determining design differences that matter in comparative human factors use. This approach is appropriate because it's been widely used in biologic research, education, and it has been previously applied in the medical field. Currently, there are 3 analysis techniques that exist to determine design differences. First, a labeling comparison, a side-by-side, line-by-line comparison of the IFU prescribing information and a description of the delivery device constituent parts. Secondarily, the comparative task analysis. It is the generic to the RLD. What are the tasks? What are they expected to do? What are the perception, cognition, and action? And then a physical comparison of the device. That includes the visual and tactile examination of the physical features, the size, the shape, the visual, the tactile sensory input that would be coming back to it. Each of these can be presented with minimal visual language. They may not be comprehensively describe the attributes of the user interface that may pose more or less risks. They say in design, devil is in the details. And it could be that the devil is in the details and using these techniques as a determinant of design differences because dependent upon the rigor of the technique, more or less design difference may be illustrated. Further, in instances where there's a known risk is better controlled by a difference in design, the guidance it could be perceived to promoting an identical rather than a safer user interface, meaning that we may be making improvements and it'll come evident, but we're not promoting that in our guidance. So what are the attributes of a user interface? In conducting a literature search, we found that there is little in it. There's little published literature about specifically user interface design attributes. What is there has an emphasis on changing customer behavior or promoting brand. As a designer, I can attest that the majority of my colleagues in design have an affinity to design, but not about writing it. However, there are a few descriptors worth noting. These include color, shape, size, texture, and material of a user interface. Seemingly, these describe physical attributes, however, they do translate into two-dimensional design. For example, the shape of a font determines if it is sans serif or sans serif. This, in turn, impacts legibility. In an FDA presentation by Wixton and LaBeouf, critical design attributes were defined as all things necessary in order to use or administer the drug product. This is a broad definition and difficult to reduce to practice to its individual elements. This lack of definition further exacerbates the situation in defining design differences of critical design elements. It isn't until there is an agreed upon language that this can be resolved. Our goal in building this taxonomy is to categorize RLDs in their various permutations, thus enabling consistent determination of design differences. We are just getting started in building this taxonomy, and I'd like to share a few examples and welcome your feedback as we journey through this complex situation. This is where we started. By taking a look from the designer's perspective, we asked the following question: what aspects of an inhaler can be variable? This enabled us to identify physical aspects of a product design, which might be important. It started us with a discussion. It opened up the doors to say, is this important? Is this not important? What is the relationship back to a task analysis? And how might these inhalers, how might use be impacted by each one of these different inhaler designs? Next, we started to gather examples of inhalers in order to develop a library. We maintained the viewpoint of simply assessing the physical embodiment. The results produce this natural organization, which is based off the fundamental shape of the device. You can see here that we have elbow design, we have a cylindrical design, a disc design, ellipse, and a rectangular design. each of them represented by several different permutations within each one of those categories. From there, we notice that upon further exploration into each device, the categorization and therefore visual taxonomy changed when we categorize based on intended purpose, medication type, and dosing regimen. What we've learned from this is that you really can't take the approach of selecting the book by its cover. As in the first example, by design, devices that look similar were actually intended for alternative medication as illustrated in this example. The taxonomy can and will change dependent upon the question it seeks to answer. We've begun assessing injection pens and known RLDs with the generics in order to better define product attributes for this important type of combination product. While the above continues to focus on the physical aspects of the two devices, we're also assessing the labeling of each. Based on our collective 60 years of experience, we know that labeling attributes and changes can look minor, but they can hugely impact performance. To date, we've generated the following possible categories for our injector library classification and have just begun to develop a robust data set. For now, our research continues. We've only started this process and will continue to explore taxonomy development, especially how it relates to use of comparative human factors. This includes considering the overall organization and categorization, which is required to make the taxonomy a useful tool for both industry practitioners as well as regulators. In addition, consideration in its relationship to other human factors processes such as the use-related risk analysis and root cause analysis will be included. I thank you for your attention today and look forward to your questions, feedback, and comments. Thanks again.